Hello, everyone. My name is Daniel Solorzano, and today I will present to you my petroleum engineering research, to which we'll focus on the topic of viscous fingering in a Hele-Shaw cell, otherwise known as the Safman-Taylor instability. Let's get into a quick overview. First, of what we'll go over today, we have the background. Next, we got the development, then the experimental method, the results, and lastly, the discussion. So what is viscous fingering? Let me explain. Viscous fingering, otherwise known as the Safman-Taylor instability, is the formation of finger-like patterns that are created when a less viscous fluid is injected into a higher viscous fluid in a porous media or in a Hele-Shaw cell. When this happens, the lesser viscous fluid tends to spread out from the injection point. That creates patterns which look like fingers. As you can see in this figure, blue-like patterns can be seen. Now, why is this important? This type of experiment shows the displacement of more viscous fluid by less viscous fluid in a porous media has been studied for many years. In many industries involving oil recovery, viscous fingering is often of great interest. One of the reasons is because of the impact it has on fluid transport. Mixing studies that can potentially yield a cost-efficient way to displace crude oil with a less costly low viscous fluid, making oil recovery efficient. Suppressing viscous fingering of an injected fluid can make oil production more efficient by a huge percentage, depending on the permeability and the reservoir conditions. Where else can we see viscous fingering? Well, viscous fingering has an impact in many other areas, such as fluid transport. The more we can study the development of these instabilities, the greater impact it would have in many other areas, such as CO2 sequestration, which is the process of injecting carbon dioxide in liquid form to displace another fluid, such as oil. Geothermal recovery. We want to recover as much thermal energy as we can. Sometimes we need to inject CO2 to displace hot brine so that hot brine can be produced. Soil and water contaminant remediation. An issue that occurs here is that in the porous media, dead-end pores trap the oil inside. Drug delivery is another area where viscous fingering can be found, and chromatographic separation. Those are some other areas as well. So this kind of phenomenon happens a lot in different areas, but our most important focus is to understand the basic fundamentals. Once we understand this, we can then work on how to control viscous fingering. There are many different forms of viscous fingering, depending on the experimental technique. It can be in a form of heat, of fluid, or even glass beads, which we will see in the next slide. The viscosity ratio is ultimately the determining ratio of to why viscous fingering develops. Changes in the viscosity ratio lead to changes of how the instability behaves. Permeability is another important factor. Fluid flow in a porous media, it can be either heterogeneous or homogeneous. For experimental study, we focus on porous media. We focus on the homogeneous porous media because of the viscosity ratio. Due to the viscosity ratio, we can still see the viscous fingering, unlike a heterogeneous porous media, where the fluid flow becomes unstable. And here are some photos. We will ultimately look at 3 experimental methods that show 3 different techniques of producing viscous fingering images. In the first figure, we can see how viscous fingering looks like using Schlieren imaging, which basically displays heat waves. In figure 2, we are looking at viscous fingering with fluid displacement. And in the third figure, viscous fingering can be seen using glass beads as a porous media that has a completely different permeability than the previous figures. The typical model that is used to study viscous fingering is the one that has a point source that has the injection point intruding the less viscous fluid at a constant flow rate into the porous media, or Hele-Shaw cell, containing the more viscous fluid that is already inside the shell. The best way to see this displacement is to use a Hele-Shaw cell with transparent glass plates. The parameters that we use is a 20 by 40 centimeter glass plate. The glass plate has a spacer with a thickness of 0.8 millimeters. The spacer acts as a sealed boundary that prevents fluid from going out. Once the cell has been properly sealed, the cell is then fully saturated with glycerol. Once this is done, the injection of a less viscous fluid can then begin at the injection point. Blue dye water enters the cell displaying viscous fingering patterns and is then produced at the production well. The injection rate is a constant and can either be high or low. In a moment, we will see the difference of a low injection rate and a higher injection rate. The production well. Production well is displacing fluid from the cell, which has less viscous fluid while it's injecting. The geometry of this experiment is said to be in a horizontal 2D flow. As this experiment is done multiple times, there are steps that need to be followed correctly to perform the experiment successfully. Explain. So in the procedure that we have the spacer that is displayed as you can see, and then we have the glass plates, once combined together, create the Hele-Shaw cell, which is then clamped together, ready for the procedure to happen. The Hele-Shaw cell is saturated with pure glycerol by putting some glycerol in a vial, which is then injected into a pump, which the pump is then used to saturate the Hele-Shaw cell, which injects a lesser viscous fluid inside the cell. If we set the distance between the two plates to be very small, the fluid flow will be in 2D motion. The fluid flow is essentially controlled by the pressure gradient between two fluids of very different dynamic viscosities. For my experiments, I use blue dyed water and glycerol. Let's take a look at what happens. This is the preparation for the cell with blue dye water, and this is the videos of the experiment, both at 2 different rates. So as you can see from the left video is slightly different than the right video. The injection rate is increasing the area of the blue fluid that is just going through the cell. The viscous fingering pattern is slower than in the one that had 1 milliliter per minute injection. The difference is also that the 0.3 milliliter injection rate showed the pattern collapsing slightly and increasing right after. The fingering pattern develops due to viscosity ratio. In these 2 cases that we saw, we use pure glycerol. The shape of the pattern tends to look just like fingers. In many cases, all look very similar while using different experimental techniques. Overall, we can study the development of these finger-like patterns to follow better way, or to find better ways to control viscous fingering and to enhance oral recovery. Thank you.